parts of the country are COVID-free now.
Okay, so good morning, everyone, and welcome to second session of International Weekly Meeting in association with CMAAO. We are on 21st of August. It's 9:30 a.m. in India, and it's raining here. And we have our guests joining in from various parts of the world. So today we will straight away start with the presentation. It is on immunology and COVID-19 learning and lessons. The data is being presented by Dr. Shashank Joshi, who is a well-known endocrinologist and physician in Mumbai, working at Lilavati Hospital. He has been past Dean of Indian College of Physicians and the past President of Association of Physicians of India, apart from several other medical societies. He has published more than 800 research papers and is actively involved in COVID-19 evidence-based work. He is an active member of Maharashtra COVID Task Force. So we will have a very well and nice data presented by one of the person who is actively involved at the field level. So Dr. Shashank Joshi, over to you for the presentation.
Thank you, Saurabh. Can you allow me to present my slides?
Yes, you can now.
Thank you. So, I thought that today when Saurabh asked me what should I talk on, I thought the most important thing today in COVID is immunology. Because, all of us know here, all over the world, that the lessons we have learned from COVID immunology are evolving. And every day we learn something new in the immunological field. And outcomes of COVID, whether they are going to be asymptomatic disease or whether somebody is going to get severe disease and die, is all linked to the host immunology. And the behavior of the virus, independent of the variant of concern, is linked to immunology. So why this topic? So the reason I chose this topic is because host immune response is the critical factor which is driving COVID, and analysis of this response allows us to get a clearer picture how host response impacts on the disease severity in some, while most infected people will show mild symptoms or no symptoms. So some people will be totally asymptomatic, some will have mild symptoms, while somebody will have severe disease. We learned a lot in the early parts when we analyzed the samples of the blood, that there is a varied interferon stimulated response and there is a HLA class 2 downregulation. We also understood that people who are on ventilator with acute respiratory B cell derived granulocyte population. And we also understood that the circulating leukocytes do not express high levels of pro-inflammatory lung storm. And we'll so obviously the study of host immune response, both in acute COVID and people who have recovered from COVID in convalescent individuals, is the key to understand the long-term protection, the early protection, long-term immune memory, and treatment measures for future outbreaks for similar viruses. Because we know that COVID is here to stay, but in order to understand better about COVID, we must understand some of these host responses in a better way. So when we all know that pathogenesis of COVID is not defined, viral research indicates that the mechanism by which COVID virus enters or invades host cell is the same. Though we don't know much about COVID origin and we are researching more and more, we know bats is a source and it's a bat related virus. That is clear cut. And human to human transmission is the key in pathogenesis. But once the SARS-CoV-2 enters the target cell, there is a spike protein association with its receptor. This we know very well now. The viral RNA is encapsulated and it is poly-identified. For sure that there is poly in it, and it encodes various structural and non-structural polypeptide genes. So we know that S and NSNPs, the genes get encoded, and this is something which we all know very well. All these polyproteins are cleaved by proteases, which are like scissors, which exhibit basically chymotrypsin-like activity. So we know the major protease in the COVID activation is the TMP-RSS2, which is a transmembrane serine protease. And this is the primary protease, if you ask me, and has been linked to SARS-CoV-2 activation. And clearly, we have now single-cell RNA sequencing data to show that it is the ACE2 and the TMP-RSS2 which are not expressed in the same cells. And also, we know that there are other proteases like cathepsin B and L, which are also in this process. We also know now we have a lot of computational biology, and using computational biology, we can viral track the various sequences and sequencing data of the viral RNAs. And this, we know that this approach was identified in bronchial. Know that viral track identified co-infections of monocytes within the human metapneumovirus. So obviously, when we look at the whole immunopathology of COVID, and this is a busy slide, I don't want you to look into it. All it tells us is that some people will have a full-fledged ARDS, which will kill people. Some people will have a completely normal response. Some people will get fever, cough, dyspnea. Some people will get inflammatory clusters with fibrinolytic material. But one thing is clear-cut, that there is a transcriptional footprint of SARS-CoV infection. It's a very distinct footprint, which is very different from other coronaviruses and common respiratory viruses like IB, HP, IV3, RSV, etc. Also, we know that there is a blunted interferon 1 and 3 response. And this signature of chemokine is very consistent. So two things we know very clearly in COVID immunology, that SARS-CoV-2 has a very clear transcriptional footprint, and it has a very consistent chemokine signature in which the type 1 and type 3 interferon is blunted or reduced. So when we look at clinical parameters and signature biomarkers in immunopathology of COVID, we know COVID immunity interval, clear signature of various, these reflect viral load and lung injury, and therefore that inflammatory cytokine signature continue to do that. And we also know that it's not just B cells, it's also T cells, which are equally important in COVID biology. So this is a very standard slide. All of you all have seen it. I'm just repeating it. Most of the time in the 80-20 may differ, but now we know that it's around 80% will have an adequate immune response, which will be timely. The innate and adaptive immunity will quickly mount a type 1 interferon response, which through macrophage mechanisms will efficiently have an antiviral response. There will be activation of Th1 and B cells, making neutralizing antibody. So that's going to be the standard 80% immune response, which is adequate. Unfortunately, comorbidities, age, or sometimes even a variant of concern may blunt this immune response and will have an inadequate immune response, where there could be a delayed or limited type 1 interferon response, which will lead to endothelial cell death, epithelial and endothelial leakage, overactivation and exhaustion of T cells and NK cells, and of course, a cytokine storm which will come in. So we have learned from the early days of the pandemic that when we look at the various predictors of mortality, we know that there are several lab parameters which are involved in cardiorespiratory failure. And from those early data till date, we have started learning the dynamic immune signature and its links with the poor prognosis, whether it is the various interleukins which are protective, for example, IL-6, IL-10, or CRP and ferritin, or the D dimers, the simple lymphocyte counts, the simple IgM spike proteins, or simple T cell counts. And based on that, initially what they did in China is they mapped the symptoms, they mapped survivor and non-survivor cohorts, and they mapped signatures. And from that, they basically came out with the first risk stratification. So this is very early data, which showed that age and male sex clearly had a hazard ratio which was significant. And then everything else was linked to the simple lab findings of white cell immunology. So clearly we understood 80% of the immune response, 15 to 20% was severe, and 1 to 2% was fatal. And this data has not changed over the last 1.5 years. The first stage of COVID is clearly asymptomatic. There is innate immune activation, there is viral engagement of PAMs, and a very low type 1 interferon response. The stage 2 is non-severe and symptomatic. There is adaptive immune activation, there is generation of specific antibodies and T cell response, and there is a release of DAMs. And finally, the 3C syndrome sets in stage 3 in a small percentage of people, where there is cytokine release of IL-1, IL-6, TNF-alpha, GM-CSF, interferon gamma, coagulopathy, and complement response. So clearly, we now know that the dysregulated immune response for COVID-19 can be predicted, and we can understand it better, and we understand the immunopathology of it better. Clearly, we also know the virus better. And we also know that we have now access to antibodies. We know that the maximum viral load is a week prior to actual diagnosis. Then we know that likelihood of PCR positivity in the first 10 days, it can get extended if in severe cases. And obviously antibodies start climbing up, with IgM coming up as early as 3 to 4 days and IgG coming up a little later. But remember that every virus has different antibodies. For example, we need to understand that whenever there's a viral infection, there are different isotopes, different targets, different specificities. For example, we have 3 types of fundamental antibodies. One is the high affinity antibodies, which are generated with maturation of somatic rearrangements and hypermutation of IgG genes. Then we have the non-neutralizing antibodies, which recognize the viral epitopes, but they do not. And then we have proper neutralizing antibody, which not only recognize the viral epitope, but they eliminate or greatly diminish the infective virus, which is critical for preventing reinfection. And the development of non-neutralizing antibodies typically precedes the development of neutralizing antibodies. But it is the neutralizing antibodies which will confer fundamentally the immunological memory. So if you look at the functional activity of immunological antibody isotypes, we need to understand that there are these big 5: the neutralizing capability, the opsonization capability, the sensitization of the antibody to natural killer cell-like killings, the sensitization of the antibody to mast cells, and the complement activation. We predominantly know that IgM, which is made, has a little neutralizing and opsonizing capability, does not have any activity for sensitizing of the NK cells and mast cells, and usually is linked to the complement activation. IgD is not very relevant. We know IgG has Ig1, 2, 3, and 4. Then, of course, IgA has a huge role in neutralization. So when one is developing a sterilizing vaccine, and none of our current vaccines are sterilizing, IgA will have a critical role. And of course, we have the other types of IgM antibodies. So each type of antibody is relevant, but 2 particular antibodies of relevance for the COVID space are Ig1, Ig3, and IgA as far as protection is concerned. Look at the time course of adaptive immunity in SARS-CoV-2. Whenever there is a SARS-CoV-2 infection, there is an innate immune response. 4 types of immune cells which are activated. One is the Th helper cells, which make cytokines. Second is the T regulator cells, which make the regulation of inflammation. Third is, of course, the cytotoxic T cells, which are equally important. And then, of course, are the follicular helper cells, which induce antibodies. And fundamentally, once there is this adaptive immunity, plasma cells and antibodies meld, we have the memory B cells and the memory T cells. So clearly, we get an immune memory. And this is a classical immune timetable of SARS-CoV-2. When it comes to T cells, as I said, B cells are important, but the induction of SARS-CoV specific memory T cells are equally important for long-term protection. We know that the T follicular helper cells indicate maturation of the humoral immune response and establishment of a pool of specific memory B cells readily to respond whenever there's a reinfection. That's the key That's the key which we have used and exploited by vaccinologists. All our current vaccines are early generation, but the next generation vaccinology will look at even sterilizing immune response. We know the specific CD4 T cells are important to elicit B cell responses and that result in antibody affinity maturation, and spike specific T cells correlate with IgG and IgA titers. We all know that we need a robust immune response with spike specific neutralizing antibodies, memory B cells, and circulating T follicular helper cells. And this is something if we see from people who have recovered from COVID infection, naturally, we will look at it more closely. I will also discuss hybrid immunity at the end of my presentation. So what happens fundamentally? Whenever there is an exposure of human being to coronavirus, the white cell counts will vary between leukopenia, leukocytosis, and lymphopenia. Lymphopenia is the most common. And we clearly know that lymphopenia and lower lymphocyte count means poor prediction. Any lymphocyte count with 800 cells reduce the chances of survival. We have even seen cases with 0 lymphocyte counts. And we clearly know that the lymphopenia with SARS has a direct linkage. There is a direct effect of SARS-CoV-2 virus on the T cell apoptosis. So that's something which is very well recognized. So let us look at each of these cells in terms of the host defense. So first is the role of dendritic cells, the ones which get trapped. The role of dendritic cells in host defense of COVID-19 is not very clear. During infection with SARS-CoV-2, the APC, that is the antigen presenting cell function, is altered. And the impaired dendritic cell migration results in reduced priming of the T cells. So that's all we know, that during SARS-CoV-2 infection, there is an alteration of dendritic cell migration and alteration of the antigen presenting cells, and therefore the priming of T cells is weaker. And this leads to fewer number of virus specific T cells within the lungs. All we are trying to do is sequestrate the lungs with less virus specific T cells. After initial infection with the virus, the lung resident respiratory dendritic cells, which are called as RDCs, seek out the invading pathogen or antigens from the infected epithelial cells. And when they are activated, they migrate to draining from the draining lymph nodes, which are called as DLNs, which are either mediastinal lymph nodes or cervical lymph nodes. And once the draining lymph nodes, the respiratory dendritic cells present the processed antigen in form of a complex, which is a MHC peptide complex, to the native circulating T cells. And then, of course, there is an engagement of the T cell receptor with this peptide MHC complex, and co-stimulatory signals induce T cell activation, proliferation, and migration to the site of infection. So this is a typical way dendritic cells play a role in host defense. What about cytotoxic T cells and NK cells? Well, we all know that whenever there is any viral infection, cytotoxic lymphocytes and natural killer cells are important, and for the functional exhaustion of cytotoxic lymphocytes are linked to severity of disease. So in SARS-CoV-2 also and in COVID-19, the total number of NK and CTLs are decreased, and that is parallel to the exhaustion of their function and upregulation of the NK inhibitory receptor CD94, the NK group 2 member A. And whenever people recover from SARS-CoV-2, the NK cells and CD8 cells are restored with their reduced expression. So clearly we know this better and we understand this better. What about CD4, CD8 and HLA? Well, we know whenever there is T cell apoptosis, which is seen initially with MERS patients and now with COVID patients, there is decreased number of CD4 and CD8 cells in the peripheral bloods, particularly in people who have HLA-DR, CD4 and CD38. Clearly, which is seen that these double positive cells and activated cells have impaired activation. And whenever there is appearance of CD25, 28, 69 expression in T cell subsets, we know that there is a delayed development of adaptive immune response and prolonged virus clearance. So now we understand little insights from immunology. Some people don't clear the virus and get a little severe infection. There may be some links with HLA. There may be some links with CD25, CD25, 28, CD69 expression on the T lymphocyte subset. We are understanding this a little better and better. The humoral response we know is very clear that lesser are the T cells, more strong is the correlation with the severity of the acute phase of SARS. So whether it is S or N protein of SARS, the immunogenic epitopes, which are recognized by CD4 and CD8 T cells, we clearly know that in order to produce neutralizing antibodies, it is important that the viral antigen is recognized by the antigen presenting cells. And this is something which we know. And we know that IgM is produced as early as 3 to 6 days, and IgG can be detected as early as 8 days. So understanding the immune response is very critical for vaccine development. We all know that the HLA class 1, class 2 epitope pools are used to detect CD4, CD8 T cells in 170% of convalescent plasma. And we know that CD4 response of SARS-CoV-2 spike protein is clearly correlated with the magnitude of the antiviral immunoglobulin titers. Though the T cell responses are also found against M, N, and open viral proteins. But what is very interesting is 40 to 60% of non-SARS-CoV-2 exposed individuals also possess CD4 positive cell responses against SARS-CoV-2, indicating that there is clear-cut cross-reactivity between coronaviruses. But this is something which we are understanding better. We also know both cell-mediated and humoral-mediated defense is important, but we know that there are a lot of effector cytokines. And in the effector cytokines, interferon gamma directly inhibits viral replication and enhances antigen presentation. In fact, people have even tried interferon as a therapeutic modality in early COVID, and we also know it may work in some trials. We also clearly know that SARS-CoV-2, there has been a lot of postulate that it secretes a novel short protein which is encoded by ORF3b, and this inhibits expression of the interferon beta and enhances viral pathogenicity. So some people feel that there is a rapid decline in a particular case, while other cases don't decline. This has been linked to probably the more virulent virus, to the novel short protein ORF3b. We also know that the cytotoxic molecules are produced by activated T cells, and various such cytokines are linked to pro-inflammatory cytokines. So clearly we now know that post-immune response is the key. Who will be asymptomatic, who will get severe disease. And we clearly know that there is a varied interferon stimulated response, and there's a clear link with class HLA2 downregulation. So clearly studying host immune response, both in acute COVID and people who have recovered from COVID, which is convalescent individuals, provide molecular insights to understand better about protection and long-term immune memory for designing various therapies. So let us quickly look at what are the markers which have evolved from all the immunological insights we got. Early days, this was early paper last year in May, which showed that these were the big five. If they are present, there is severe COVID likelihood is more. Like neutrophil to lymphocyte ratio. If it is 5.5 versus 3.2, severity is likely to be more. D-dimer, if it is high, it is likely to be more severe. CRP, IL-6, LDH, and ferritin. All these are linked to more severe COVID. And this was the data of survivors and non-survivors, originally coming from the Wuhan cohort in China and subsequently replicated all over the world. Now, what do we understand? We understand that if there is leukocytosis and neutrophil count which goes up, yes, there is a link. We clearly know lymphocytes go down, platelet count goes down, NK cell counts go down. So from the hematological front, those are the abnormalities. We also understand that the NLR, MLR is equally important, but we understand that biochemical markers like low albumin, elevated AHC, OT, SGPT, bilirubin, myoglobulins can also be important. Coagulopathies and D-dimer, fibrinogen are equally important. And inflammatory biomarkers, including cytokines like IL-2, IL-6, IL-8, IL-10, TNF-alpha, ferritin, CRP are equally important. And these are all the links and the data which has been published all over the world. And now we know this process very well. It's a thromboinflammatory disease. Whenever there is SARS-CoV-2, there is a dual pathway. One is the activation of the coagulation system, and other is the storm of cytokines. And obviously, whether it is the inflammatory cells or the mononuclear cells, through various pathways, there will be the activated platelets, there will be the impairment of the natural anticoagulant pathways and shutdown of fibrinolysis. But many factors impact disease. whether it is the viral load, the immunological dysfunction, radiological parameters, and so on and so forth. And there are many scores which have been developed now to predict the severity of COVID. But the key summary is this slide which I'm putting out here. That whenever the SARS-CoV-2 is trapped in the airway epithelial cell, you have two types of responses. One is a cellular response and second is a humoral response. In the T cell and NK cell response, which is the cellular response, clearly we know that you will get a very efficient clearance of the virus. And in the B cell response, the plasma cells will ensure that there will be some degree of complements as well as antibodies which are produced. And of course, the dendritic cells will make the antigen presentation better. And if the interferon response, cytokine response has low levels, most people will recover. While if the levels of cytokine go up, you will get a cytokine storm which will impact the heart and the lungs. So clearly now we understand this pathway better. And interferon has a key important role in the respiratory tract and severity of COVID. In fact, the interferon landscape is now better understood local or regionally. And the interferon expression of COVID varies with location, varies with viral load, varies with age, and varies with severity. We clearly know interferon gamma 1 and 3 drive protective, immune stimulating in the mild upper airways. So in mildly ill patients, if you have good interferon which is made, most people in the upper respiratory tract, it will be nose and throat COVID, and there won't be any lung COVID. However, the critical patients may express interferon alpha beta or gamma 2 with low immunoglobulins and high P53 expression. And the epithelial cells might express some of these. So clearly, when there is severe COVID, there is overproduction of immune mediators, and the role of interferons, whether it is 1 or 3, is still debated. However, we clearly know that the production of interferons along the respiratory tract in COVID patient has been found to be high, particularly for the interferon 3 and the upper respiratory airways of people with high viral burden, but that is linked with reduced disease risk and severity. So clearly we know that when there is mild pathology, our body makes a lot of specific interferon 3, not interferon 1, and probably that efficiently drives the transcription of genes which protect us against SARS-CoV-2. In contrast, people with other infections or non-infectious lung pathologies, interferons are overrepresented in the lower airways with severe COVID, and then you get worse disease. So there is a dynamic production of interferons in SARS-CoV-2 infected patients, and they show that interferons play an opposing role based on the anatomical sites. So the role of interferon in the upper airway and the role of interferon in apoptosis in the lower airway or the lung is clearly linked. Let us look at the last part of my presentation, which is fundamentally the cytokine release syndrome. Because the big key question which we know in immunology is the cytokine release syndrome. We clearly know that normally when we get viral infection, we get a normal cytokine response to viral infection. For example, the inhaled virus will infect the lungs. The virus will be detected by monocytes and macrophages. These monocytes and macrophages will release IL-6 and other pro-inflammatory cytokines. These cytokines will recruit T cells, which will stimulate pro-inflammatory cytokine responses and magnifying cascade. These cytokines will promote vasodilation and make leaky capillaries. And of course, then the lung will be full with fluid, leukocytes, and inflammation. What is happening with cytokine release syndrome is the recruited T cells fundamentally have the hyper amplification of the pro-inflammatory cytokine response. And whenever there is a recruited T cells, there is a hyper amplification of the pro-inflammatory cytokine response. Clearly, we know that the SARS-CoV-2 will infect and destroy the T lymphocytes. There will be lymphopenia, and the extra or overwhelming viral load will magnify the inflammatory response until all the lymphocytes will be exhausted. So that's what happens when there is cytokine storm, and that is typical of that. So in the cytokine release syndrome, there are 3 immunological stages: stage 1, stage 2, and stage 3. Stage 1 of the immunopathology of cytokine storm is initiation. where there is early induction of predominant cytokines and viral sepsis, where the ferritin goes up and lymphopenia is seen. There is infection-induced chemokines, then and they begin to recruit innate and adaptive immune cells to the lung lesions. Stage 2 is amplification, where there is large production of lot of inflammatory cells, massive recruitment of inflammatory cells, including pathogenic T cells, which amplify the immunopathogenic process. And the stage 3 is the consummation stage, where there is continuous inflammation, markers of inflammation, and widespread organ damage. So these are the 3 stages. Usually the first stage of initiation is 4 days. Usually the amplification is from day 5 to day 14, and consummation is day 15 or death. So this is how we know and we understand COVID cytokine storm better in various stages. T cells are the key. In the innate and adaptive immune response of COVID, we clearly now know that the SARS coronavirus infects macrophages. Macrophages present the SARS-CoV antigens to the T cells. This process leads the T cells to be activated and differentiated. They make cytokines with different T cell subsets, including Th17, and then there is a big release and virtually a diarrhea of cytokines for the immune response amplification. However, the continued production of these mediators due to viral persistence have a negative impact on the NK cell and CD8 T cell activation. And the CD8 T cells produce very effective mediators to actually clear the SARS-CoV-2 virus. So clearly, T cells have a role in activation, T cells have a role in exhaustion, and lymphopenia is a consequence. So whenever there is a decrease in the peripheral blood T cells, it is associated with severity and inflammation. That is very well documented. We also know activated CD8 and CD4 cells display a trend towards exhausted phenotype, and that is based on what we understand on the cytotoxicity and the polyfunctionality. So clearly what happens now is the 3 things which are illustrated in this diagram. The first is there is depletion and exhaustion of lymphocytes. So when the ACE2 mediated entry of COVID enters into lymphocytes, a lot of pro-inflammatory cytokines and the cells are exhausted and depleted, and the SARS-CoV-2 virus will damage lymphatic organs, causing lymphopenia. And the lactic acid levels will inhibit proliferation of more lymphocytes. So that is the first thing, a lymphopenia, exhaustion of lymphocytes, and depletion. The second is increased neutrophils. Lymphopenia may be secondary to a microbial infection, but there is clearly a neutrophil activation and recruitment, which happens. Third is, of course, the mechanism of cytokine storm induction. Usually a Th1 signal is activated that secretes a GM-CSF with further induction of monocytes with high IL-6 levels. Whenever there is an induction of monocytes and macrophages, they promote the IL-1 beta, and the Th17 cells which produce IL-17 recruit more monocytes, macrophages, and neutrophils. And that is the key stimulator of the cytokine cascade of 1 beta and IL-6. And finally, and the last thing, which might have a role futurely in vaccinology, is the ADE, antibody dependent enhancement. We know that the neutralizing monoclonal antibody targeting the virus enhances virus entry into the cells via the FC region of the antibody bound to the FC receptor. So you can clearly see that this is what happens here, that this is the cell, this is the viral entry, and you can see that the antibody will target the virus and it will enhance the further viral entry and it will launch a vicious cascade. So clearly some people will go through the hypoxia, pneumonia, mechanical ventilator, while some people who will have a robust humoral response will have a different pathway. So this is typically how multi-organ failure will occur. The T and NK cells will get exhausted. Some people might get secondary uncontrolled infection and die, and some people might recover. That's typically the COVID pathway. We know that the CD6 has a co-stimulatory receptor cell on the T cells. And we know that people have now used even a humanized anti-CD4 monoclonal antibody, which is like itolizumab, in the T cell proliferation. And we know that these are T cell modulators which are used, and they have different actions on T cell modulation. And they also express the Th17 and Th1 cytokines. So obviously we know that we have various immunological targets which we can target with various substances like cytokine response modulators, and so on and so forth. So we have a lot to understand on the T interactions. So clearly now to block the cytokine storms, we have three types of agents which have been very useful across the world. One and the most popular one is the anti-IL-6 monoclonal antibody, which is tocilizumab, that binds to the IL-6 receptor, blocks the IL-6 mediated signaling to immune cells. The immune modulation of the T regulator cells is not modulated. It clearly blocks the IL-6 alone. It's short duration, so sometimes we need to give 2 doses of this. But current data on the tocilizumab is quite exciting, and it has shown that it may have a life-saving potential if timed appropriately. So if your patient is deteriorating on steroids, despite of adequate oxygenation and prone position, tocilizumab may be able to save lives. There are other 2 agents which have also been equally useful. One of them is itolizumab. It binds to the CD6 receptor, blocks the ALCAM mediated T cell activation, has a clear role on regulatory T cells, downregulates, and has a longer duration of action due to upstream pathways. So you can give it much earlier. And we also have the anti-IL-1 receptor antagonist like anakinra, which blocks IL-1 receptor, blocks IL-1 signaling alone, but has a shorter duration of action. So these are the 3 agents which are useful. So which one is useful? Well, tocilizumab is a cytokine receptor blocker, while itolizumab is a cytokine formation blocker. We have both of them available, and both of them have a clear role. One, the formation blocker can be used early in the disease, while the receptor blocker can be used right up to even ventilated stages. So this whole immunomodulators and targets are huge, whether it is steroids, whether it's IL-6 blockers, IL-1 blockers, GM-CSF, anti-TNF blockers. So many trials, including the recovery trials, are undergoing. And you can see the number of agents being investigated all over the world in this space. Well, let me come to the last part of my presentation as we conclude, that the main thing which we want to understand today is the immune response against coronavirus. We all know that immunity to severe SARS-CoV-2 is a vital issue for global society today. And determining the quality and duration of immunity is the key. So the acute part of the COVID we dealt with, the cytokine storm we dealt with, but what is key to us today for survival of planet Earth, for the 7 billion people living in the world, is the quality and duration of immunity. And that's really going to be the key. But our adaptive immune system is very complicated. And obviously, there are clear-cut factors. When you get SARS-CoV-2 through natural infection, the immune response mounted by natural immunity versus vaccine-generated immunity is also going to be different. So this is the key for understanding, as we understand how much are we going to be protected and what type of immunity we have. So therefore, a concept has been now evolved called hybrid immunity. Like most people were talking in the good old days called herd immunity. But today, what we are going to get on planet Earth is hybrid immunity, which means a question of combination. What type of immunity will develop in people with natural immunity who are subsequently vaccinated? That hybrid immunity is particularly interesting because the notable finding is people with previous SARS-CoV-2 infection mount unusually potent immune response to COVID vaccines. So they say that people who have had COVID and have taken a COVID vaccine are more likely to be better protected than people who are just taking a single vaccine. And if people who take 2 types of vaccines with mix and match probably will have a little better immune response because it's a heterologous immune boost. So remember, natural infection with vaccine probably is the best example of hybrid immunity. So this was a science paper which I was alluding to, which was recently published. This is called as the hybrid vigor immunity with COVID vaccines. And that is where we know the first tree is natural immunity, second tree is the vaccine immunity, and the third tree is the hybrid immunity. And you can very clearly see that the different plant lines are bred together. The hybrid is much more stronger. Something similar happens when natural immunity is combined with vaccine generated immunity. There is a 25 to 100 times higher antibody response driven by memory B cells and CD4 T cells. And there is a broad cross protection from variants, because today we are also concerned with variants. So here you can see the different shades of blue, the memory B cells, the antibodies, the CD4 T cells, the CD8 T cells. Because all we are doing is measuring sometimes antibody as a response to a vaccine. But we are not looking at the memory B cells. We are not looking at the CD4 T cells and the CD4, CD8 T cells. So clearly we know that we understand better about this hybrid immunity better. Finally, we are all here because we want to live in a world with immunological memory. And we know that for protective immunity, immunological memory is the key. So we have two types and paths of protection. One is natural immunity and second is vaccine generated immunity. We know that the adaptive immune system consists of three branches. One is of course the B cells, which is a source of antibodies. Second is the CD8 T cells and third is CD4 T cells. For natural immunity, now we have clear data to suggest that after natural SARS-CoV-2 infection, there is global published data to suggest that immunological memory to SARS-CoV-2 is seen for more than 8 months for CD4 T cells, CD8 T cells, memory B cells, and antibody, with a relatively gradual decline, which appears to be partially stabilizing within a year. So for 8 months to 12 months, clearly you can see here that you will have some form of a natural protection. The level of immunity can be placed on a spectrum, and the natural immunity against the symptomatic COVID infection has been found to be 93 to 100% over 7 to 8 months in large studies, including locations where there have been variants of concern like alpha, which was widespread. So this is also strain independent to a great extent. So obviously we are concerned that what type of natural immunity is seen against variants. Will it blunt reduce the production of antibody recognition? For example, if there's a beta strain like B1351 or the P1 gamma or the iota or the delta, it is less clear. And we don't have so much of evidence against such variants. But what is clear is that the neutralizing antibody activity against most variants of concern is reduced for natural immunity and vaccine generated immunity. So the most of the vaccine or the variants of concerns have mutations engendering the partial antibody escape, because there is a selection pressure to evade natural immunity. So the biological relevance of reduction in neutralizing antibody potency against variants is most clearly evident from vaccine trials and observational studies. You have seen what has happened in Israel, and you have seen what is happening in people who have got 2 vaccines and what is happening with the breakthrough infections. In Israel, we know more than 70% of the population was vaccinated with the Pfizer vaccine. Still, we saw that there was some degree of selection pressure, and we saw that there was clearly some degree of an antibody escape, particularly against the Delta variant. So this is going to be relevant as we go by. So currently among the various vaccines, whether it's Astra vaccine, where we know that efficacy of symptomatic cases dropped from 75 to 11% against the South African strain. Similarly, for the Pfizer, the symptomatic cases dropped from 95 to 75% against the South African strain. So remember that vaccine efficacy is very, very peculiar, and we do not know whether it is coming up or not. So obviously, hybrid immunity is a concept which is impressively very potent. There is a clear synergy observed between antibody response more than the T cell response after vaccination, and its enhanced antibody response depends on the memory T cells. There is a clear discordance which is there, which means that hybrid natural vaccine immunity will be reproducible to enhance immunity. For example, we know Shingrix vaccine will prevent shingles when given to people previously infected with varicella zoster virus, is impressively effective in 95% and elicits a very high antibody response than the viral infection alone. But these principles can also apply for COVID vaccine modalities. So currently the thinking is that if we use 2 different kinds of vaccine, the heterologous prime boost regimen can elicit substantially stronger immune response than either modality alone, depending on the order in which they are used and on what vaccine modalities are combined. But this is all in research mode. So clearly we know that many combinations are going to be tried, and some trials have been published, whether they are the mRNA and the adenovector viral platforms or the mRNA and the recombinant protein vaccines. Clearly we know that the SARS-CoV-2 immunology has thrown a lot of pleasant surprises. And this can potentially be leveraged to generate better immunity for COVID-19. And that's something which is evolving every day. Thank you for a patient hearing.
Thank you, Dr. Shashank. Wonderful data and very in-depth, detailed analysis. We will quickly move with the introduction of the associations. I just want to mention that we have about 40 people over here, and CMAAO is a representation of 19 countries: Australia, Bangladesh, Cambodia, Hong Kong, India, Indonesia, Japan, Korea, Macau, Malaysia, Myanmar, Nepal, New Zealand, Pakistan, Philippines, Singapore, Sri Lanka, Taiwan, and Thailand. We have representatives over here, and the whole idea is to make this as a think tank and bring the collective information for during this pandemic. I would not. I would like to, also acknowledge the presence of our chair, Dr. Chong, immediate past president, Dr. Ravi, Vice President CMAAO, Professor Ashraf, Dr. Elvin from Hong Kong Medical Association, also the treasurer for CMAAO. We have Dr. Pillai from India and member for World Medical Council. Dr. Salma from Pakistan Medical Association, she's the president. Dr. Angelique from South Africa Medical Association. Dr. Jamal Chaudhary from Bangladesh Medical Association. Dr. Akhtar Hussain, again from South Africa Medical Association. We have Dr. Sajjad from Pakistan Medical Association, Dr. Marie from Japan Medical Association, Dr. Russell from Australia UNESCO, our presenter, Dr. Shashank Joshi from India, Dr. Bukhari from Pakistan, Dr. Monica Vasudev from United States of America, Dr. Sanchita from IGCP Group India, and I'm the host. Also, I would like to ask if I have missed out, because I've seen a lot of people over here joining in. So have I missed out on any association name over here who has been representing and we have not acknowledged the name? Kindly introduce yourself, please. Just unmute and say your name, and then we can start with the country update. Anybody representing any association which we have missed out over here? Okay, over to Mr. Chong for...
Dr. Prakash is there. Dr. Prakash, you can say.
Dr. Prakash.
Mm-hmm.
Okay, so Dr. Prakash, and he's from Nepal. Okay, yeah, I missed out his name. Sorry, Dr. Prakash. Yeah, Dr. Chong, over to you.
Thank you, Sarab, and thank you, Dr. Joshi, for such a brilliant, presentation. Really good summary of the situation. I think there's some questions in the chat that I've seen. Perhaps Dr. Joshi can answer them. I see some questions there.
Hello, Mr. Chairman, this is Dr. Kaisar Sajjad.
Yes.
Okay, can I ask? It was a very good presentation, Dr. Joshi, and I missed the beginning of the presentation, but I just have a question that the WHO says that the mix and match vaccine is not good for the human being, and your presentation says that the mixed vaccine is very good for to build the immunity. In Pakistan, we have mostly the Chinese vaccine, Sinopharm and Sinovac, and the third vaccine, which is being recommended as a booster dose, is advised to be vaccinated with the other vaccine like Pfizer and Moderna. So what do you think that it's a good strategy to have a one vaccine of the other group and the other vaccine is the other group? Number one. Number two, do you think that these vaccine will be continued as we have the other vaccine like flu vaccine every year, or what do you have any suggestion in this regard?
Doctor, these are very good questions, and I think you make 2-3 very relevant points. So let's not discuss about WHO here. They will have policies based on equity of vaccines and distribution of vaccines. So public health policies and individual policies often differ. Remember, when you do a public health policy, it is for most of the people, most of the times. Clear data emerging all over the world now is that heterologous prime boost or a natural infection followed by a vaccination of different platforms will have better immune response. So that data globally is very clear cut. Whether it happened because of serendipity, whether it is occurred because of a design of a trial, that data is clear cut. The second thing which is evolved is, yes, in the future we will have mix and match, but we are in this field an emerging field. So we are generating science and data on this. Number three is hybrid immunity is also here to stay, because we will have a large population in the world which will be exposed to the virus, both symptomatic and asymptomatic, and then we'll be giving them vaccines. So that also is like a immune boost. So in all these situations, one thing is clear cut, that if you use the same platform and give 2 or 3 shots, the antibody response which will be generated versus if you have a natural infection with antibodies followed by a vaccine, the response is going to be different. And if you use 2 different vaccines, the heterologous prime boost will have a more immune response. So that data is clear cut, doctor. So, and it will evolve. Country to country, it depends on the availability of the vaccine. Currently, the booster is recommended only for immunocompromised people. In absence of immunocompromised states, booster is not recommended. There has been a global debate that there are 7 billion people in the world, and only the rich are using boosters and for immunocompetent people, while there are people, you can't eradicate COVID till we have the last mile. So that is the global debate happening. But currently it is prudent to say what you said, Dr. that we need to understand and evolve country-specific strategies of mix and match. For example, in the city of Gorakhpur in India, accidentally people got Covaxin followed by Covishield. The immune response was better. That was published by the ICMR. Similarly, all over the world, Spanish study, UK study, US studies have shown that whenever mixing and matching has occurred with different platforms, and your own experience from Pakistan, you have shown that that there is going to be a better immune response. So scientifically speaking and immunologically speaking, when you combine different vaccines, the heterologous prime boosting will have a more robust immune response and a little long lasting immunity. That is what has been the conclusion based on the published data.
Okay, thank you.
Is this the data you're talking about?
That's correct. That's correct.
Yeah, so I can send this link over this.
Thank you, Saurabh.
But there is a difference of opinion between the WHO and the US. USA is only planning to go for booster for all, actually. So what is the justification of this decision?
So Dr. Jamal, it's a very sensitive and a political issue. WHO is looking at, as I said, from a global perspective, a public health perspective, most of the time for most of the people, everybody should get at least 2 vaccine shots and at least get some degree of vaccine-induced immunity. That is what.
Yeah, but that doesn't justify actually the US decision if it is depending on the level of antibody level.
US.
Then why WHO is telling that when the people are not getting the vaccines and the US and those who can afford, they are going for 3rd vaccine. So that doesn't justify actually. Only thing is that scientific evidence regarding the antibody level will justify. It is actually the efficiency and the affordability of the world actually, I must say, as a whole, that they must produce enough vaccines. It's not that you can just say that when people are not getting vaccines, so you are going for that. No, it's not like that.
So I agree with you completely.
Yeah.
From a scientific standpoint, for immunocompetent people, booster is a questionable entity. We need better science. For immunocompromised people, you must give a booster dose. Clearly, yes. Answer is yes. Number 2, I don't have very high respect for WHO. With due respect to WHO for 2 reasons. Number 1, they are the same people before March last year who said that this is not an airborne disease and don't even wear a mask. So I really don't have much of, and they have not been guided by science. So I think let us not get into that controversy. Let us be guided by science. The science is very clear cut. You see, we have Asian experience, whether it is Singapore to Japan, which has clearly shown that masking is the way to go. And I think when WHO said that last year in February and March, I think it was appalling. Having said that, there were aerosol and airborne scientists who had to push them and they acknowledge even their mistake after such a long time. What you are saying is very true. World should have adequate vaccines for every human being on planet Earth. That's number one, accepted. Number two, what is the science telling us? Science very clearly tells us that you must vaccinate everybody in the eligible population in the eligible doses. And after that, if there is not sufficient protection, which are those groups which are likely not to get protection from vaccine? The immunocompromised group. Whether somebody has a cancer, somebody has kidney failure, transplantation. And for them, a booster is recommended. The Center for Disease Control, Atlanta, Georgia, has clearly suggested that for immunocompromised people, you must give a booster. For immunocompetent people, they are thinking of giving a booster because of the Delta strain. Because they have observed that with the Delta strain, Dr. Jamal, what you said is very correct, that the antibody response is not very good. It has dropped. Because of the drop of the antibody response, they are justified in saying that let us give a booster. But the ethical debate on that is we must give vaccine to everybody first and then probably look at a booster. But the exception to that is immunocompromised people. I think immunocompromised people deserve the booster. Immunocompetent people, whether they deserve the booster is depending on affordability, accessibility, and the local regional geography and policy decision. So that is not our scope. Our scope is to discuss science. Our scope is to discuss what is correct. And the science is very clear that if you are immunocompromised or if you have not mounted a robust immune response after adequate vaccination, which you call quote unquote adequate, we must take a booster. Very clear answer, yes.
But Dr. Joshi, actually, in US, actually, it has been seen in case of Delta, it is a 75% effective even still now. So why they are going for third, for the booster? 75 is also good.
As I said, that US FDA has not approved the booster dose outside the scope of immunocompromised states. And I am not,
But they are going for by September.
That is their.
Yes.
We cannot comment on what they do. We can only do what we can do for our country. And I think we have to look at our countries and our decisions. I personally feel they have some logic in because of the Delta. See, two things have come. Even in the US, after 2 Pfizer vaccines, they are seeing outbreaks. Even outbreaks, including hospitalizations after 2 vaccinations. So they feel the need for doing a booster, even in immunocompetent people after 6 months to 1 year. They will have data on that. That data may not be in public domain all the times. So I think let us look at what is the science evolving. Immunocompromised states, no debate. Yes, give it. Immunocompetent people, likelihood of breakthrough after 6 months or 1 year is there. That is why somebody else asked a question, will you need an annual dose? Yes, probably, like flu viruses. We may need a dose, but we don't know whether it's going to be every year, every 8 or 9 months, every 7 months, we don't know. Because the strains keep on changing. When the vaccines were made, there was no Delta, Alpha or Gamma. Now there is Alpha, Delta and Gamma. So obviously booster is needed. So yes, but from 95% efficacy, it has come to 65% and 75% efficacy. So there is a blunting. Look at what has happened with Israel. Israel is fully vaccinated and still they are having breakthrough infections and they have seen a surge. So obviously they have justified their booster dose. So each country will make their own geographic decisions based on what is available, what is accessible, and what is best for their population. So I don't think we should comment on what is there. We have to focus on how to protect our country, our people to the best of what we can do.
We have a lot of questions coming up and we have to move on with the questions.
Dr. Joshi, I just add on this what Joshi is saying, that the immunocompromised people should have the booster dose, as you said. But what about the healthcare workers? They are very much vulnerable. What do you suggest for the healthcare workers?
So the Indian Medical Association has clearly asked for a booster dose. Dr. Martan Pillai, our representative, is here, and Dr. Jailal, they have clearly written to the government that healthcare workers and frontline workers should be given a booster.
Should be given, yeah.
Okay. In Pakistan, the Pakistani government is also thinking and announced the same thing. Yes.
Absolutely correct.
It's okay. Thank you.
Angelica, you want to ask a question?
Yeah, thank you so much. Very interesting discussion. I'm just coming more from a clinical point. Dr. Joshi, my immunology are not very good. I always realize it when I listen to these type of meetings, and I thank you for helping. just want to know regarding the pathology lab test type of thing. When do you decide, or in what part of the disease progression would you want to do your full blood count to double check whether this patient will be is going to be compromised or not? Do you do it when you make the diagnosis? Do you do it 5 or 6 days later or 10 days later? Or do you look at your clinical picture if the patient is progressing and getting worse? When will you start? Because again, not everyone can afford these tests, but I know it can add a lot of value. If you can just help me on that, thanks.
So it's a very good question. Ideally, there is a dynamic process in the 14-day timetable of COVID. And usually we start doing these tests from day 5 to day 10, because that is the time that the patient can deteriorate. Ideally, if you can do a CBC, CRP every day or every alternate day, every 48 hours, it is good enough. You get a direction. So that is what we do normally. But if you can't do it, then do it on day 5, day 10, you will get a reasonable thing. And then, of course, monitor it with saturation or a pulse oximeter. But if you really want to do the test, sometimes we do it daily or alternate day. CBC and CRP is good enough. You don't need everything. Ferritin picks up very early. IL-6 may not be very good as a assay. D-dimer is not needed in everybody. So we know what to do and when to do. But your question is very relevant, that if nothing is available, do it on day 5, day 10. If you can do it alternate day, great, between day 5 to day 12.
Thank you, Dr. Joshi. Excellent. Any questions from the floor? There are some questions in the chat box, Dr. Joshi.
Yes, so Dr. Chan has asked a question that the spraying nasopharyngeal vaccine, nasal vaccines are going to be sterilizing vaccines. See, all the COVID vaccines which are available today only prevent death and severe disease, but do not prevent COVID infection. So you will get a breakthrough. While the nasal vaccine will be a sterilizing vaccine, which means you will not get COVID. So that's a very good question which doctor has asked. The other question, Dr. Chong, you had asked about the ADC, ADE, that body dependent enhancement associated with dengue vaccine has not been classically seen with SARS-CoV-2, but that was a concern. You know, Lutz Mentenegger, who got the Nobel Prize for discovering HIV/AIDS virus, expressed a concern that these vaccines might put too much of immune pressure and might actually explode and cause ADE. But all the vaccine trials have included ADA, and all the vaccine manufacturers have given a commitment that ADE is not a problem with the SARS-CoV-2 virus. Maybe in the dengue space, there was clearly the likelihood of the ADE. So that's something which we need to understand and we need to do better. Then again, you know, are the self-replicating RNA vaccines being developed? And do you think that they will have longer immune memory, or will side effects from cytokines release be worse? This is a very difficult question to answer, Dr. Chong, because we don't know. There is a risk that it may deteriorate. So I think biologically there is a possibility and plausibility of deterioration, and therefore we are not sure whether really we'll get self-replicating RNA vaccines. But in India now, we have a good news. Yesterday we approved a DNA vaccine, and the DNA vaccine was approved even for children between 12 to 18 years of age. So that is something which is very unique. So we'll get more and more vaccine options. Diane is asking a question. What about the safety of exosome 24? Well, exosome 24 has its own pros and cons, and exosome 24 has been well known, you know, for in the COVID space. But currently it is more in an experimental mode. So offline, I'll be happy to send you literature on exosome, you know, 24 and COVID-19.
There is another question in the direct.
Dr. Joshi,
Yeah.
Joshi, can I just, although I don't know whether it will be relevant or not, I'm not getting any information regarding the Covaxin approval from WHO. Covax, because it is related to Bangladesh also. So actually, because Covax came here for a clinical trial.
On that,
I'm sorry?
it is in active process. You know, it is more of a bureaucratic process, and by first or second week of September, we'll get that approval in place.
Thank you.
It's a matter of time. I think all the vaccines will get their approval, and it is just a matter of time. So as far as the exosome is concerned, we all know about exosome, that it is a soluble CD24, and it's nothing but a CD24 has a huge role in the T cell population. And when we give that CD24, it negatively regulates inflammation and suppresses cytokine storm. That is where they have tried the trial. It's undergoing trials right now. It is undergoing feasibility trials, CD24 exosomes for moderate to severe COVID disease. And predominantly, it has been tried in Israel, and some other countries might also take it up.
There's another question.
Dr. Joshi.
Yeah.
Yeah, this is Dr. Alvin Chan from Hong Kong. Thank you very much for the, can you hear me?
Yes, Dr.
Yeah, okay, thank you. I have two questions. Thank you very much for your excellent lecture. Though I had joined late and I could not listen to the first part, and because of my clinical duties today, even on Saturday morning. Well, one question is about the now under research nasopharyngeal spray vaccine. I don't know. Now, is that because the virus first entered the body through the nasal pharynx and the mucous membrane, and so they think that the IgA or the local reaction there, I mean the immunity there should be an advantage. But I like to know, say your expert view. And the second question is a general strategy choice question. So in mainland China, in some places, they are aiming at a zero case approach. So they would have to aim at a zero case eruption. So Hong Kong also is now following this strategy. But of course, another strategy is in an opposite direction, is to live with the virus. We have to accept that this will be the coming scenario, is an endemic thing, and annually there will be variants like the influenza. So we will have to approach this, and then life will go on, economy will come back, etc., etc. So these are two different strategies giving to opposite directions. So what are your expert views upon, a global aspect? The world should go on, but which strategy for which country should be more advantageous? Thank you.
Dr. Chan, you asked two very good questions. The first question is on the nasopharyngeal vaccine. Including India, there are 4 global manufacturers undergoing trials for sterilizing vaccines. It is called as sterilizing antibody. You're very right. The portal of entry for COVID is through the nose, and therefore nasal spray is the right way to have a sterilizing vaccine. In India, we have a company, Covaxin. We are undertaking the nasal vaccine for Bharat Biotech, and there are 3 other manufacturers undergoing trials, including in China. These trials are undergoing for nasal vaccine. So they are called as sterilizing vaccines. So these are ongoing trials. Even we are involved in phase 2, phase 3 trials of the nasal vaccine in India. Second is a more important question. Will COVID be endemic, or can COVID be eradicated? Can we have a zero COVID free world? And will it become like MERS and SARS-1, extinct? Unlikely. If you ask my question, it is all hypothesis. Your guess is as good as mine. It is unlikely that like MERS and like SARS-CoV-1, SARS-CoV-2 will be eradicated. There will always be some pocket in the world somewhere where you will get a flare-up. Therefore, there will be some degree of endemicity. So we will have to live with the virus. We will have to take our viral vaccines, and we will have to probably, like our flu shot, we will have to keep taking it. Now remember, Spanish flu started in 1918, but till date we are not getting flu peaks. But it has become endemic in our population. Some geographies have a lot of flu, like influenza season, like Northern Hemisphere, North America and Europe, while our countries don't get so much of flu. But that is what will happen with COVID also. We will all live with COVID. but we will get some degree of immunity, and that is my hypothesis. I don't think we will be able to have a zero COVID time. I wish that happens, that there is no COVID at all and we have no more discussion, but it looks unlikely now. If it had to happen, probably, the third wave, fourth wave, fifth wave would not have occurred. You in Hong Kong, you have seen, Singapore, you have seen, every time you have lifted the restriction, there has been a spurt. Every time you have allowed people to come from outside, there has been infection. And we are putting too much of pressure. So we might get more and more variants also of COVID. Because this is a slowly mutating virus. Every 7 days, the virus is mutating. So I think we are putting too much of selection pressure. And my worry is that we should not get a more deadly strain. We should not get a more deadly strain. I think the Delta itself was a deadly strain, but if we put more pressure and do too much of vaccination, and that is where Dr. Jamal was asking a question, WHO also has some scientists which are skeptical of too much of vaccination. If you put too much of pressure on the strain, there might be a more deadlier strain coming out. So I think it's important that we will have this virus endemic. I think by next year, the disruptions due to COVID will stop. So right now we are disrupted. Many parts of the world, there are lockdowns. I think world will be more easily mobile place after a year. That's what my feeling is. But of course, this is all speculative.
Thank you, Dr. Joshi. Very good summary of life after COVID, or rather dealing with post-pandemic COVID world. Yes, I think this is the way to go. I mean, there's really, it's going to be very difficult for a zero tolerance COVID policy. Although I'm sure China and Hong Kong will pursue it, but it's not going to be easy. So any other questions from the floor?
Otherwise, Dr. Chong, we have 15 minutes. We can give 2 minutes to everyone this time. Otherwise, we'll skip the other meeting.
Okay, we'll try our best. Let's do a quick summary of what's happening. Alvin, you want to go first?
Thank you, Chairman.
I would just request everyone to spend 2 minutes on the update this time. We just
Thank you. Yeah. Wow, so many things happening. So we have to just squeeze. Well, of course, in Hong Kong, it's still quite good. We have just 1 or 2 community confirmed cases amid hundreds of Delta strains entering Hong Kong from overseas. But just those 1 or 2 cases will scare people to death, And because Hong Kong is following China's approach to have the zero tolerance approach. So, it's difficult. Well, this is an international meeting, so I could speak out very freely, of course. But if in Hong Kong, we are still speaking out freely, okay? But you are in danger of being attacked sometimes now. Even in mainland China, a scholar from Shanghai, he was saying that, we should live with the virus. That Dr. Zhang had been attacked all over China, internet. Okay, that is really dangerous. Make us think about Dr. Li Wenliang again, the whistleblower last year's January, and then who's penalized and then who died, and then he became a hero, of course. But then in Hong Kong, I think we are in a difficult time because when we have to have a zero approach and not to live with it, then even when Nicole Coleman, coming into Hong Kong to have the TV shooting, the legislators and some KOLs just yelled and said, "Oh, that's not equal," and that's a fuss, saying Hong Kong is so endangered by these celebrities, etc. So I think it's creating a fuss. So at the end of the day, I agree with Dr. Joshi that globally, you can't really have a zero tolerance approach. In a country or in a city, you might have that approach, but you are not an isolated island in the world. So I don't think that is quite practicable in a way. But then, of course, we will have to face another thing. I think I have to raise it here. that say we have to always now say that this is Hong Kong SAR of China, and Macau is Macau SAR of China, and to mainland China, okay, Taiwan is Taipei, China. Okay, so in the next month meeting, I think I will have to be very careful. Okay, I will have to give anything verbally or orally like this. And eventually, the Taipei statement has something that if we don't agree, say technically, like telemedicine in Hong Kong, our professional conduct handbook still think this is not really ethical enough if we have universal practice like that. And then we could not agree. So that part of the statement, if we don't agree, then we could not agree with the whole statement. And that would be easier for me to perform, you know. That's how Hong Kong thinks. Okay, do you understand, Chairman?
I understand. Thank you. Let's move on to Ravi from Malaysia. Ravi, anything to tell us?
Oh yeah, plenty to tell actually. We are in the midst of our cases rising. We had 23,000 plus cases yesterday, 233 deaths yesterday, which hovering between 200 and 300 deaths. In the midst of a political turmoil, which I'm sure you would have read about or heard about. As of today, we have a new Prime Minister will be taking office at 2 o'clock Malaysian time today. And of course, it's the same group. The Deputy Prime Minister is becoming the Prime Minister. Of course, he will probably have a new cabinet. Just now, I was asked a question, what about the public sector? The public sector, of course, remains the same. It's only the ministerial level we will have changes. So everything is proceeding as normal. The country at the moment is concentrating on vaccination. We have actually vaccinated about 29.8 million people, out of which 17 million have got 2 doses. Though our population is about 37 million, including children and everybody, we have approximately 4 to 5 million migrant workers, documented and undocumented. So that is the group that we are very concerned about. However, what the government is now doing is they have emphasized on vaccination and they're going all out to vaccinate. Our Director General of Health has come up lately and said that the load on hospitals is reducing, on ICU and those on ventilators is reducing. So they say. But otherwise, the number of cases seem to be increasing. So this is our concern. Amidst all this COVID-19, we have a political turmoil in this country to add on to this situation. So people are very, very anxious, very concerned here that a time like this, there should be no political turmoil. There should be concentration on how we can control the COVID-19. So this is scenario in Malaysia at the moment.
Thank you. Thank you, Ravi. Maybe I'll move on to Angelique. Angelique and Dr. Hussein, whether you'd like to update us on what's happening in South Africa?
Thank you so much. I will leave the numbers to Dr. Hussein. I just want to say, to make 2 brief comments. I feel for Ravi and I feel for Dr. Chan, because I think politically, politics should never be a part of health. And unfortunately, it is even so in our own country. So just want to say that we have now finally given the green light to 18 years and above to all be vaccinated. We will see how it is playing out. I think one of the reasons that was done is pressure that has been put on the government, but also now we have a very low uptake on vaccines, lots of vaccine hesitancy in our country. And I am afraid that, you know, although our numbers are still 13,000 plus per day, that it feels to me that we are becoming complacent. You know, it is as, okay, and what's new anymore? I think people are just carry on with their lives, don't look at and listen anymore. And that might be a problem going forward, especially around vaccination and trying not to distribute the virus. But I'll leave the numbers to Dr. Hussein And just want again to thank you all for this very important Saturday morning meetings. It helps a lot. Thanks.
Thank you.
Thank you.
Dr. Hussain.
Thank you, Dr. Chong, and thank you, Dr. Joshi, for excellent presentation. The number in South Africa last 24 hours, 14,312. Those are the new cases. And our death last 24 hours, 289. So far, total, we have tested more than 15 million. And the positive cases out of 15 million is 2.6 million. Recovery rate is 90.7%, and positivity rate is still 21.6. We had countrywide ventilation till yesterday, about 1,200 patients, and total ICU admission yesterday was 2,400.
Wow.
Thank you, Chair.
Thank you, Dr. Hussain. Sorry to hear these figures. Can I have Dr. Jamal to tell us about what's happening in Bangladesh? Dr. Jamal?
Dr. Jamal, you need to unmute yourself.
Sorry. Okay. Thank you, Chair. Actually, Bangladesh was under lockdown one week back, but that was actually the cases were rising. Actually, now the day actually it is open, it was just come down to 50% of the 16,000 to 8,000 like that. But surprisingly, in these 7 days, after 7 days of this opening up, it has come down still more. So we are expecting, and our advisory committee was just expressing their concern that it will increase more, but it is getting down. I don't know why. It has come down to near about 5,000 like that. So less than 5,000, and the number of death cases also has gone down actually. So Bangladesh is going for vaccination, and they are trying to open up the schools, universities, academic institutions. So they are allowing the students to get vaccinated as early as possible. And this is the scenario actually in Bangladesh. Thank you.
Thank you, Dr. Jamal. Is Dr. Marie around? Sorry, I think I missed out on her. Is Dr. Marie around?
Hello.
Yes,
Hello.
hi, Marie.
Hi. Thank you very much for every week. In Japan, it's getting better compared with last week, but we have still many patients waiting for the hospitalization, and some of die during their home evacuation. So that's too bad, especially the 30s and 40s are the victim of this fifth wave. We are rushing to have the vaccination all ages, and the 65 years old and older has vaccinated around 80%, but the younger, including the younger generation, there are only 50% is vaccinated right now. What is happening right now in Japan is like this. Thank you very much for every week.
Thank you, Marie. Can I have, is Dr. Salma around? If not, can Dr. Sajjad or Dr. Bukhari update us on what's happening in Pakistan? Dr. Sajjad or Dr. Bukhari?
Assalamu alaikum. I think Dr. Salma is there.
Is she around?
Hello? Can you hear me? Hello?
Anyhow, in Pakistan,
Dr.
the
Sajjad
situation is...
Hello? Yes, yes. Hello? Can you hear me?
Yes, Dr. Sajjad.
Dr. Sajjad, yes.
Okay. Thank you very much. Actually, in Pakistan, we last week, our president, Dr. Salma, told that the fourth waves are in progress, and the number of cases was increasing, particularly in one of the province in Pakistan, which is in Sindh, and in Karachi, particularly the cases was very much increasing, and the positivity rate was 30% in Karachi. and gradually it is increasing all over the Pakistan, in Punjab province and the KPK also. But now from the last 1 week, it's going down. The percentage of the number of cases and the death is going down. And today, only 3,087 people are infected within 24 hours, and only 57 people have been died till now. So we hope for the better progress in Pakistan in near future, and the vaccine. People are very much enthusiastic, and they are going to the vaccination centers and get and get themselves vaccinated. So we hope that in the end of this year, the 50% of the population will be vaccinated in Pakistan, hopefully.
Thank you, Dr. Sajjad. Thank you. Dr. Bukhari, anything to add?
I think we can quickly move on with the other sessions.
Yeah, okay. Dr. Pillai, anything to update us? On
Thank you. Indian situation is definitely improving. Still, we have a caseload of about 35,000 per day. But then this is a little fallacious because the cases are mainly from 2 states, which contribute to almost 75 to 80%. So generally, we are getting things under control, and the R value, which is better than the TPR rate, which obviously is 16, R value is 0.89. If it is less than 1, that means things are under control. So this is true of almost all the states, even those states where the number is high. We have almost tested the PCR test and the COVID test we have done for 50 crore of people so far. So that is a great number. And vaccination, 44 crores have got first dose and 12 crores have got second dose. That means 30% first dose and 12% second dose. That is where we stand. And the happy news is that India has produced the second new vaccine. That is a plasmid DNA-based vaccine, which has undergone even the third trial. And the third trial, 28,000 people have been tested, and the efficacy is 68%. And now this is open for vaccination. The drug controller has approved it. Another good aspect of this vaccine is that you don't need needles. It is not intramuscular, it is a intradermal. So that you just, without a needle, it is applied. So that the local reaction, adverse effect will be much less than the intramuscular injection. So that is a significant achievement that we have got, and that is where we stand. Thank you.
That is some good news, Dr. Pillai. Dr. Joshi, do you want to update us as well?
No, I think I agree with Dr. Pillai that we have a new vaccine now. It's a 6th vaccine approved in India. Second thing is we clearly having clear cases declining, except for the location of Kerala and Maharashtra. Everywhere else the decline is there. And even in these 2 states, things are much better under control. The R value is below unity, and the second wave is on the decline. So overall, India looks reassuring, and let us hope and pray that next genomic sequencing is occurring. So in Maharashtra, we had 15 new cases of Delta plus detected, but none of them had any critical threshold. So the number is less than 1% of the total genomic population. Currently, the predominant strain in India based on genomic surveillance is still the Delta. There is a sporadic sprinkle of Delta plus, but by and large, overall things are looking better.
Dr. Joshi, we have another question which is relevant for the India, and I believe a lot of stakeholders from the roundtable has also joined. The question was that India has begun crushing the Delta wave, Delta curve. What is India doing right, and can you share the experience? Like, what is India doing to make sure that the Delta variant is not moving out now?
No, viruses comes in waves, so they come and go. But India is doing their simple strategy of they have continued the high testing. So despite of the low prevalence of test positivity rate, we have maintained high testing rates. So we have crossed 50 crores as testing. So the testing is there. So we have persisted with testing, tracking, and isolating. So I think that is the key. So if you need to really contain the pandemic, we need to keep testing, testing, and testing. And with that, you will see the test positivity rate gradually coming down. And eventually we'll see that. Also, we are vaccinating a lot. We have more than 55 crore Indians who have already got one vaccine shot. India is the largest vaccinator on planet Earth right now. No other country has given more vaccines with the exception of China. Because China, we don't have data, but probably they have vaccinated their whole country. After China, probably India is the largest country in the world which has given 55 crore vaccines. So it is vaccination and testing, which is the key, and sound public health policies.
Thank you, Dr. Joshi. So in Singapore, I just want to update everyone. We've hit about 82% of the population with the first dose, 77% with 2 doses. But as Dr. Joshi has mentioned, we are moving away from border controls, social restrictions, to vaccination and public health measures like tracing, swabbing, testing, etc. So I think that is the way to go, as opposed to, let's say, a zero tolerance policy. So in fact, Singapore and Hong Kong wanted to do a travel bubble, but because of the differing views, we have decided to call it off. So, or in a way, it's called off. So that's probably the way to go that we are headed towards. Okay, I think we've come to the end of the meeting, and
we can just cover the last topic for the US, since Dr. Monica is still awake.
Yes,
I would take a privilege for her to speak something what is happening in US with
Yes,
the end
please.
of the session after that. Dr.
Yes.
Monica, please.
Hi, thank you. I won't take much of your time. I did post the updates regarding the 3rd dose on the group chat regarding the guidance that we had received within our institution. I do know that there has been conversation regarding the 3rd dose for everybody. However, I think the focus is getting scheduled the people who are immunocompromised. We are well aware of the equity disequilibrium, is how I'll put it, that the rest of the world has not received enough doses of the vaccine while this is being offered in the US. I just do want to make a comment, though. I think it should just be a state of wherever vaccines are available, they should be utilized and administered following the science. There's no point having stockpiles of vaccines in places that are not being used. And it also doesn't make sense to have vaccines that are available and are being refused because people feel that one is better than the other. There are a lot of discussions regarding distribution, but if a vaccine to be protective, given the scenario that is found in your local area, then I feel that we should let the vaccine be given where it should be given, and we should all continue to wear the mask. I hope that by sharing the information that I receive within the US regarding how we distribute the vaccines, that we are well aware of the comments and the emotions that go behind this. We have to still continue to do this. We know that every pandemic is going to live through its multi-year cycle. And I do believe that this will become endemic. I do predict that we will need to continue to receive these vaccines. It'll be clubbed with our seasonal influenza immunizations that we received annually for all healthcare workers and for the general population. And as these requirements increase, the availability of vaccines will be increased. The spread in different varieties of vaccines will increase, as we've seen with the new DNA vaccine, which I will highlight is 3 doses. It's transdermal. It requires a process where it needs to be through a pulse through the skin. So every vaccine will have its own way, its own method of being used. And I think that we should just welcome and celebrate every vaccine that's available and continue to use them efficiently. So I will continue to share information as it comes to me. And I wish all of you the best. Thank you.
Thank you, Dr. Monica.
Thank you, everyone. And I think the next meeting is here, so we've got to scoot off now. And
we have been the bad boys today. I think we have now taken over 10 minutes of their meeting today, this time.
Thank you, everyone. Thank you.
Thank you very much.
Thank
you. Bye-bye.
So thank you, everyone, for joining in for the wonderful meeting. And we will see you again next.
Thank you. Thank you so much.
Bye, everyone.